Hello, good afternoon. Welcome back. So far in this course, we have talked about how viruses get into cells and reproduce, and mostly we have focused on the virus. We've talked about how you need transport systems from the cell, you need polymerases and so forth, but we really haven't paid much attention to how much virus infection actually alters the cell. And that's what I want to talk about today. This is called the infected cell. It's meant to be an overview of what goes on. We can't talk about everything, of course, so I've selected a few topics to give you an idea of how when a virus infects a cell, it's really no longer a cell. It's an infected cell because now it's devoted to making new virus particles. And in many cases, not always, but in many cases, the cell will eventually die as a consequence. Now remember that everything a virus does in a cell depends on metabolism of the host. It depends on biosynthetic and signaling pathways of all different sorts, trafficking, of course. And so what I'm going to do today is give you an integrated description of what virus infection does on the host cell. So there is an infected cell. It's one of the two parts of a virus. A virus is an organism with two phases, the virus particle and the infected cell. And that infected cell has been completely changed to making virus particles. So that's what I want to give you a sense of today, how that happens in different ways. And these are the four things I want to talk about today. I want to talk about how signal transduction pathways, which are really crucial, obviously, for the life of the cell, how those are altered by virus infection. I want to tell you how virus infection alters gene expression of the cell, which includes mRNA synthesis and translation to make proteins. Of course, metabolism is impacted substantially because it takes energy to make molecules, and so if you're going to make more viruses, you're going to have to turn that up. And finally, how cellular organelles are remodeled. In cells infected with polioviruses or related viruses, after 4 hours, there are no more ER left. There are no more Golgi membranes left. It has completely been remodeled so that the virus has a place to replicate its genome. So we'll talk a little bit about that today. So let's start with signal transduction. Signal transduction pathways are essential for the life of a cell because cells have to sense what's around them, whether it be another cell or nutrients or lack thereof. They have to be able to sense it and respond. And in fact, just about everything a cell does is governed by signal transduction pathways like the one shown here. And for sure, viruses alter signaling pathways of many different types. And I want to give you an example of just a few of them today. On the left here is a very important signal pathway in the host called the PI3 kinase AKT-mTOR pathway. Now, I'm sure you're used to learning about signal transduction pathways in other courses with all the cryptic abbreviations and having to memorize them. I don't want you to memorize any signal transduction pathway. I just want you to understand the impact of virus infection on them. Now, here is the, as I said, the PI3 kinase AKT-mTOR. So here's PI3 kinase, a very important protein kinase that signals to other components of the cell. Here's AKT. The point here is that this is showing you a signaling pathway that is made to sense nutrients in the extracellular space. We have a number of receptors on the cell surface, all of which, when bound by their ligands, will turn on PI3 kinase. In this and all of these slides I'm going to show you, the green arrows mean stimulate, so an engagement. RPTK is a receptor protein tyrosine kinase. These sense growth factors in the medium, and then they start signaling. They turn on PI3K, which in turn turns on something else, et cetera, et cetera. And the red arrows, of course, mean inhibit. So these kinases, when bound by ligands, turn on PI3K, which has downstream effects of various sorts. And you can see that some of the end points here, apoptosis, programmed cell death. So if something's not right, the cell is going to die. But right here, you're seeing AKT is inhibiting all of these. So these signals, these positive signals from these receptors will inhibit cell death because they mean that, for example, with the RPT bound to its ligand, it means there's a growth factor present so the cell can remain alive. Down here are some of the other outputs, proliferation of the cell. So again, when growth factors are present, the cells can proliferate, they can grow, and they can undergo autophagy under other conditions. Right, so these pathways govern how cells respond to its environment, and viruses modify how that happens. And on this slide are shown a number of viral proteins that intercept into these pathways. You can see the viral's protein names are given next to the virus, adenovirus, E4-ORF. The name of the protein doesn't matter, but you can see all of these proteins in the upper right, they can stimulate PI3 kinase, and the downstream effect is cell growth and proliferation, which of course a virus needs to have a cell growing in order to reproduce. Some virus proteins interfere, or in this case, augment later on in the pathway through mTORC, for example. And some virus proteins inhibit. So we have VSV protein inhibiting AKT, inhibiting these proteins. We have a variety of other proteins inhibiting this complex as well. So depending on the outcome required by the virus, they can stimulate or inhibit some of these pathways. We'll look at some specific examples in a moment. But here, this is a cell growth pathway, so you can imagine that a virus will stimulate cell growth so that it's able to reproduce, and cell division, like we talked about last time. So here's a closer look at a particular kind of signaling that is involved in virus entry. So we've talked about how viruses bind to receptors and get taken into cells, but there's a lot of signaling going on in those pathways that we didn't talk about. Okay, so here are three examples. On the left is adenovirus binding to cell receptors and being taken up into the endosome. And when we talked about that, we talked about it in isolation. We didn't mention that, in fact, there are signaling pathways needed for all of that to work. So, for example, when the virus particle is taken up into the coated pit, signaling events begin to occur, shown by the green arrows, through PI3 kinase, and the RAC and yellow kinase there, RAC. I didn't actually discover that, okay. And what these do is the green arrows pointing to these little orange actin molecules. You know, underneath the plasma membrane of the cell, there's an actin microfilament structure, which gives the cell rigidity. And for endocytosis to occur, that has to be loosened up. Coated pit can't just push through it. So these signaling events, again, mediated by virus binding, loosen up the actin microfilaments, and they also stimulate endocytosis. So the green arrow there shows you that endocytosis is stimulated. So this is in response to virus binding. You can imagine that some other ligand that the cell might need also stimulates these pathways, and the virus has simply tapped into it. They haven't invented anything. So that's one example. In the middle is influenza virus. Similar story, virus binding causes clustering of receptor protein tyrosine kinases in what are called lipid rafts. These are specialized microdomains of the plasma membrane. They have different composition from anywhere other places. And so virus binding causes these proteins, these receptors, to dimerize. You can see they become activated and they start phosphorylating, including PI3 kinase, and they will activate it. And that signaling, again, loosens up the actin. It stimulates endocytosis. But there's another pathway here that's quite important. MAP kinase is activated by this dimerization of the receptor, and that upregulates the channel in the endosome membrane that pumps protons into the endosome and drops the pH. And that pH drop is what eventually will uncoat the virus by conformational changes in the hemagglutinin. So that's obviously important for virus entry, and that has evolved to facilitate this particular virus coming into the cell. And the last example is on the right. where the binding of a Ebola virus, a filovirus, to the cell surface. Remember, this is taken up by macropinocytosis or pinocytosis through endosomes. The virus binds to a fusion receptor in the endosome, and then the nucleocapsid ends up in the cytoplasm. Well, it turns out that when the virus binds on the cell surface, it initiates a signaling pathway through PI3 kinase, AKT, and RAC, and RAC blocks non-permissive vesicle taking up the virus. In other words, at this point, the virus can either go through a permissive pathway for fusion or to a non-permissive pathway for fusion. And this signaling blocks the non-permissive pathway. It keeps the virus going to the right kinds of endosomes where the fusion will occur after binding of the virus to the fusion receptor. Again, these are things we didn't point out last time, but today that's what I want to try and do, is show you how signaling and metabolic and other pathways are important for virus infection. So signaling via this particular PI3 kinase is important for virus entry. Some other things happen with this PI3 mTOR AKT pathway. There's mTOR now, which we didn't see before. Some other examples of how viral proteins can modulate a balance in the cell between survival and apoptosis in this case, right? Survival and programmed cell death. So the cell naturally has to make decisions about whether it's going to go on or whether it is not going to go on. And so this is mediated through some of these signaling pathways. So on the left, we have a number of viral proteins binding to PI3 kinase, again, which is just beneath the plasma membrane, including hep C virus, adenovirus, a rotavirus protein. And these are all stimulating PI3K and activating AKT, which will block cell death and stimulate cell survival. And AKT further signals down to mTOR, which is a key regulator of protein synthesis or translation, and that will allow translation to occur. So this makes sense, right? These viral proteins are pushing the cell to survive and to actively make more protein because viral mRNAs need to be translated. And on the right, some other examples. There's a herpes virus, HHV8, a G protein coupled receptor, which is a multi-pass transmembrane protein there on the plasma membrane, can also stimulate PI3 kinase and activate, in this case, cell proliferation through a cyclin, and again, translation via mTOR. And again, inhibition of apoptosis, because the virus will not be able to reproduce if a cell dies. Although in some cases, when reproduction is almost done, apoptosis may be beneficial for the cell to die and release virus particles. So there's always a balance between these two activities. So these are examples of how this relay can be used to influence the metabolic activities of the cell with respect to either living or dying. We've been talking a little bit about AKT, which is a kinase downstream of PI3 kinase. And here's a really interesting example where a virus RNA actually induces apoptosis, programmed cell death. And so here is a flavivirus, which is a virus with a plus-stranded RNA genome, a single RNA. The virus is enveloped, and the genome is translated to a polyprotein, which is then cleaved by proteases encoded by the virus to get the various proteins it needs, including RNA polymerase and so forth. As we'll see in a moment, cells have anti-RNA defenses. They have enzymes that chew up RNA, not just their own. Our own RNAs have to turn over. mRNAs can't last forever. They would be translated forever, and you don't need every protein all the time. And so the cell has a system of nucleases to turn over RNAs, to degrade them. And it also has a defense against viral RNAs coming into the cell. And that's what this Pac-Man looking like shape here, XRN1, is an exonuclease of the cell that's involved in cell mRNA turnover and defense against cellular viral RNAs. And what this XRN does, when the viral RNA comes into the cell, this XRN1 starts chewing it up. It chews up most of the RNA, including the open reading frame. So this little orange thing labeled ORF, that's actually the whole open reading frame. It's not really to scale here. But there is a 3' non-coding region, which you can see there. And the XRN1 chews almost all of the RNA, like 95% of the RNA, and it gets to this stem loop here, and it can't go any further. And so it's left with this small fragment of RNA, which is just a very 3' end. And this small fragment of RNA actually inactivates AKT, which promotes apoptosis. So normally, AKT blocks apoptosis. But if AKT is gone, the cell is going to enter apoptosis. So sfRNA inactivates it, and it promotes apoptosis. It acts through an anti-apoptotic protein called BCL2, but the details aren't important. The fact is that this viral RNA causes the cell to die, which you may say, that sounds weird, but it probably happens at a time when virus has already been produced, and it's just a way of getting the virus out of the cells. Now, if you make a virus mutant, which can't make this small RNA, this sfRNA, it's easy to make these mutants, which would allow the XRN1 to chew up the whole 3' end. And that virus will not form plaques. Here's a plaque assay on the right. The wild-type virus makes nice plaques, but if you take away the ability to make this small RNA, that virus can't form plaques. That virus will still replicate. In fact, in cells, it produces good amounts of virus, so you're not taking away the ability to replicate. You're simply taking away the ability to form plaques, and that is because you need apoptosis, cell death, to form a plaque, at least in this case. Again, why this would happen is not clear. I would say it has to do with virus release from cells, and the plaque assay probably supports that. If you take that mutant virus where it can't make the sfRNA and you put it in mice, so normally these viruses will cause disease in mice. This virus lacking the sfRNA doesn't cause disease in mice. So apparently you need programmed cell death to not only make plaques, but to cause disease in mice. So that's an interesting example of how the cell is trying to degrade the viral genome. It's left with this little RNA, which then makes the cell enter apoptosis. Now, you may be thinking, how does the virus replicate if the RNA is all degraded? Well, not all of it is degraded, just some of it. Obviously, viruses have antagonists of enzymes like XRN1, and so this virus is still able to replicate, but enough small fragmented RNA is made that it has this effect. Let's talk about gene expression. And you remember this from a couple of lectures ago. This is transcription and RNA processing. We have a double-stranded DNA in the nucleus. It's transcribed by Pol II. It's spliced, forming a capped polyadenylated RNA, which is then exported. And this whole pathway is interfered at almost every level by various viruses. So one virus doesn't inhibit at every step, but collectively everything is inhibited. All these red letters are where different viral proteins interfere in the various steps in this pathway. So for example, these particular polio, VSV, and alpha virus proteins interfere with Pol II transcription. And I note here, polio also inhibits Pol 1 and 3. It inhibits all three polymerases. And the mechanism, by the way, it degrades the transcription factors that are needed. It doesn't touch the enzyme. It just degrades the transcription factors. Polyadenylation and splicing are inhibited by viral proteins, and RNA export is also inhibited by a number of viral proteins. Translation is inhibited by viral proteins, as we'll see in a moment. And there are some viral proteins that actually degrade the RNA, as I said before. Not only does the cell have enzymes to degrade RNAs, but viruses encode them as well. You might imagine that, okay, some of these DNA viruses, herpes simplex viruses and adenoviruses, those are DNA viruses that are in the nucleus, maybe it makes sense that these would interfere, but why would an RNA virus interfere with transcription? Well, of course, it's to reduce the competition for translation. If you make a lot of cellular mRNAs, these are going to compete for the ribosomes and the translation apparatus, and the viruses have to compete as well. So they eliminate as much cellular mRNA as they can so that their mRNAs have access to the ribosome. Remember, every virus has to make mRNA that can be translated. That's one step in common. They have to undergo protein synthesis. And so by eliminating cellular mRNAs, as all of these strategies do, you eliminate competition for the translational machinery. Here's a look in a little more detail at inhibition of export and splicing. So this is a slide that I showed you before without all the viral proteins on it. So at the top here we have a primary transcript of a mRNA with an intron that has to be spliced out. And this is being bound by the spliceosome. So all these ribonucleoproteins proteins are bound to the RNA. And splicing occurs in the next step there, and the intron is removed. There are viral proteins that can interfere with splicing, like a herpes simplex virus protein called ICP27 will interfere with splicing. So again, that's to make, if you interfere with splicing, that transcript doesn't get out of the nucleus. Remember, splicing marks it for export, so that will keep it in the nucleus, so less competition for viral mRNAs. And then there's a whole host of viral proteins that are interfering with export of these mRNAs from the nucleus. We have here on the lower left, polio and rhinoviruses. They have proteases encoded in their genomes that cleave NUPs. VSV also encodes proteins that interfere with export as well as influenza virus and adenovirus. Adenovirus actually has a big complex of proteins that form. Viral proteins are in red, and the others are all cellular. The other proteins here are members of the ubiquitin family. So this is going to target the nuclear pore proteins for ubiquitination and eventual degradation. So it basically inhibits export as well. So again, a major focus on keeping cellular RNAs in the nucleus. Of course, if you're a DNA virus, you have to be careful about what you export, right? The adenovirus has to be able to let its own RNAs out and not cellular RNAs. It has to be selective. As I said earlier, the cell regulates its own mRNAs because you can't have every RNA around forever. Some of them last only minutes and some of them last hours, and that's controlled by sequences in the RNA and a series of RNA binding proteins. So here you have a typical mRNA capped with an open reading frame, poly A tail. The way these are degraded starts with deadenylation. There's a protein with this very long name that chews away the poly A tail. And when a mRNA lacks the poly A tail, then the rest of the RNA degradation machinery of the cell recognizes it and it will chew it up. And you can have 5' to 3' decay or 3' to 5' decay. And the name means exactly that. It can be chewed in from the 5' end. And here, so this mRNA on the left has been deadenylated, so it's now marked to enter the degradation system. And two things can happen. It can have the cap removed, and these DCPs, that stands for decapping proteins or decapping enzymes, they're going to remove the cap. There you go. Off it goes. And then once the cap is removed, XRN comes in and chews it up, because that's the signal for XRN. No cap on this RNA, we're going to get rid of it. And the same way, when a message has been deadenylated, it could also enter 3' to 5' decay, where an exosome here, a purple chewer, comes in and chews away, and it's helped by another enzyme called DCPS. So these are ways that cells have to modulate the half-lives of their own mRNAs, and of course, they can also affect the presence of viral mRNAs, and so therefore, these proteins have to be antagonized or degraded, otherwise the viral mRNAs will be removed. And one example is in polio-infected cells, PAN, DCP1A, and XRN, which are all chewing enzymes shown on this slide, are all degraded. And many viruses will antagonize these pathways. Otherwise, their mRNAs will not be around and infection will stop. So this is a defense against RNA, but it can be overcome. Actually, it's always an arms race. One is fighting against the other, and who wins in the end is the outcome of a pretty interesting, delicate balance. So I just told you about cell proteins that degrade RNA, but many viral genomes encode proteins that degrade cellular RNAs. Again, to get rid of cellular RNAs, to have all the ribosomes to you. And here are a bunch of them on this slide. And the viral proteins are shown in red. So SARS coronavirus, for example, encodes a protein that is brought to mRNAs by, it recognizes ribosomes that are translating it, and then it chews up the mRNA shown there in the second slide. And then once the chewing has started, that chewed RNA is recognized by cellular XRN1, which takes away the rest of the task. So the NSP1 chewing basically removes the cap, and that brings in XRN1. Here we have a couple of others, a herpes simplex virus protein and an HHV8 protein, which recognize the cap binding complex, a series of proteins that bind the cap during translation, and they can initiate decapping, and that also leads to XRN1 degradation. And finally, vaccinia virus actually encodes its own decapping proteins that they don't just nick the RNA as do the VHS and SOX proteins there. They actually take off the cap, and of course that sends it to the XRN pathway as well. So the bottom line here is that the virus can encode enzymes that will chew up cellular mRNA. So not only do the viruses interfere with mRNA biogenesis, but if any mRNAs get out of the cell, they'll be chewed up by this system. Now here's a really interesting example of a virus mechanism for resisting some of this being chewed up by the cell enzymes by using a cell RNA. And the virus here is hepatitis C virus, which we will come back to many times from now on. This is an RNA virus that infects the liver, as the name says. And here's a schematic of the genome on the upper left here. The open reading frame is shown in orange and purple boxes. And what's at the 5' end is a highly structured RNA that's very important for replication and translation. As you can see, there are a lot of stem loops there. And let's take a look at that very 5' stem loop. It's exposed down below here. And this stem loop is bound in the liver, in liver cells specifically, by two microRNAs, microRNA 122. So miR122 binds at two places in this very 5' stem loop. And that protects the viral RNA from degradation by cell enzymes, in particular XRN1. So a cellular microRNA is protecting the viral genome from degradation by a cellular nuclease. It's amazing, right? This miR122 is only found in liver cells. It has a role in cholesterol regulation. And that's why the virus only replicates in liver cells. It's hepatotropic, and it's because of this miR122. In other cells, if it happened to get in, the virus happened to get in, it would be degraded by XRN1. This has therapeutic value. People have devised what they call an antagomere. You get it? Antagonist of miR. It's an RNA that binds the miR122, and it binds really tightly and prevents it from attaching to the viral RNA, and basically the RNA is cleared. So you can take a person with hep C infection who's chronically infected for years, you could give them this antagomere, and it will cure their infection. So this has been in clinical trials. We'll talk about this later when we talk about antivirals. So that's a nice example of how there's an interplay between cellular and viral degradation mechanisms. Our first question, which of the following is a consequence of viral proteins modifying signal transduction pathways to promote replication? Poliovirus inhibition of transcription by RNA polymerase II, herpes simplex virus protein blocking pre-mRNA splicing, disruption of actin filaments to allow endocytosis, initiation of mRNA degradation by viral proteins. Well, the answer, as you can see, is C, disruption of actin filaments to allow endocytosis. That's a case where signaling, the virus binds a receptor, initiates signaling through PI3 kinase, disrupts actin filaments. The polio inhibition of transcription is just the viral protease cleaving transcription factors. The blocking of pre-mRNA splicing, same thing, no signaling involved, neither in the initiation of RNA degradation. None of those had any signaling components. In fact, we can go back and look at some of them. No signaling here, just cleavage of RNA by enzymes, same thing here. This is all cleavage, no signaling. And here on this slide, these are all viral proteins that cleave other things to prevent either transcription or splicing or export. So the only one that involves signaling is here, the pathways to virus uptake. That the virus binding initiates signaling pathways that loosen up the actin microfilaments. Let's talk about translation and how it's regulated in response to virus infection, because you might imagine that viruses would regulate it, right? Because it's a key point where all viral mRNAs have to be translated. And remember, from the Baltimore scheme, every virus, all our 7 classes of viral genomes all come to mRNA that has to be translated on ribosomes. So you can bet that viruses are going to mess with the cellular translation apparatus. So here's how translation occurs on most messenger RNAs. On the top is a messenger RNA in green with a cap. There's the AUG codon where translation is going to begin. And the way translation begins is the cap recruits to the RNA a series of proteins that bring the ribosomal 40S subunit to the RNA. So you can see EIF4E is a cap binding protein. It binds to the cap specifically. That's why you need a cap for efficient translation. EIF4E binds to another protein called EIF4G, and that in turn binds to EIF3, which binds the 40S subunit. So you can see a series of cap protein ribosome interactions bring the initiation complex to the RNA, and then the ribosome moves down the RNA until it finds an AUG codon and then initiates translation. When an AUG codon is found, there are a series of these initiation proteins are released. The initiator tRNA is placed at the initiation codon. So when that tRNA, which comes down with the initiation complex, when it sees the AUG, you have a series of reactions that occur, and the 60S subunit will then come in, and that will signal the initiation of translation. So now you have the first tRNA placed there with its met amino acid on it. So that is what we call cap-dependent or 5' end-dependent translation, because it won't start unless you have a cap at the 5' end of the RNA. Now, you can bet that viruses muck with this, and there's some really, really neat things that they do. Here is an example of what one virus, poliovirus, does to cell translation. So here's a graph of the rate of protein synthesis at different times after infection. This happens to be poliovirus, and you can see in uninfected cells, the rate of protein synthesis remains more or less the same under these conditions. In poliovirus-infected cells, it's almost zero after 2 hours post-infection, and then it goes back up again. And what this is cellular protein synthesis is inhibited by virus infection, and then it's replaced by virus translation of viral proteins. And on the right, you can see the same experiment, except that we've labeled proteins with S35 methionine, and we fractionate them on the polyacrylamide gel. And we're looking at time post-infection, 0, 1, 3, 5, and 7 hours. You can see lots of cellular proteins made at 0 and 1 hours There are thousands of cellular proteins. And then look, at 3 hours, you start to see the cellular proteins go down and they're replaced by what are viral proteins. 5 hours, very little cellular proteins made. So that's the shutoff of host protein synthesis by virus infection. And many, many viruses do this. Why? To get all the ribosomes for their own mRNAs, right? To get rid of all the cellular RNAs on the ribosomes and replace it with their own. So how does that happen? There are a number of ways that have been sorted out for this selective inhibition of translation of cellular mRNAs. You can't do something that's going to trash the entire translation system, right? Because then viral mRNAs won't be translated. And they're shown here, a number of different viruses. The one I really like is shown at the top, and it involves cleavage of EIF4G, which is that linker protein. Here's the cap binding protein, EIF4E. So look at the bottom here. The cap binding protein binds the cap, and it brings the ribosome via these protein-protein interactions. These viruses encode a protease that cleaves EIF4G at that jagged line, and it separates away the cap binding protein. But these viruses don't need it. Poliovirus is not capped. It binds to the ribosome in a different way, as we'll see. So this is fine. This shuts off all capped mRNA translation, but the viral translation continues. You can also have regulation of translation by phosphorylation. The cap binding protein is best when it's phosphorylated. It can be dephosphorylated by viruses as well. And finally, the third mechanism involves a cell protein called 4EBP, which is a regulator of translation. 4EBP will bind the cap and inhibit translation only when it is dephosphorylated. So in its phosphorylated state, 4EBP will not interfere with translation. And this is dephosphorylated by a number of viral proteins. So a number of ways that you can interfere with cellular translation specifically. Now, why does viral translation still occur? So here are some examples. At the top is what I've told you is 5' end-dependent initiation of translation. The ribosome is brought to the mRNA via interaction of this cap-binding protein with the cap, and that then brings around all the other proteins. So that obviously cleavage of 4G there at the jagged site blocks all that translation. Many viruses do what's called internal initiation. The ribosome actually binds internally. It doesn't need a 5' cap, and in fact, these RNAs are not capped. It just requires EIF4G to recognize the structure of the RNA, and it will bring the ribosome in. Hepatitis C virus RNA has an extreme version of this where the 4DS will actually bind directly to the 5' end of the RNA. So this gets around the need for a cap, and that's why these viruses can interfere with cap-independent translation. The ability of these viruses to bind the ribosome or EIF4G internally is called internal ribosome entry or internal initiation. And so here's the poliovirus RNA genome. At its very 5' end, there's a long untranslated region of about 740 bases, which is shown expanded at the top there. There's the first AUG that initiates translation, but all this other RNA is highly structured, and that's where the ribosome binds to get the initiation of translation. There's no cap at the 5' end here. There's only a VPG protein which is left over from RNA priming. And so these RNAs at the 5' end are very structured, as I said. Here are 5 different types of these structured region. Here's, for example, the poliovirus region. 5' end is shown in each case, and you can see lots of stem loop structures. And what we think happens is that these structures recruit, in the case of these viruses, EIF4G, directly without the need for all those other proteins. You don't need a cap. 4G binds directly to the RNA, and for the hepatitis C virus, it's in the upper right here, hepatitis C virus, the ribosome can actually bind directly to this structured RNA. Again, no cap needed. So these are called internal ribosome entry sites or irises. All right, so that's one way to abrogate competition with the host cell mRNA for translation. Translation, though, is highly regulated at other levels, and I want to show you one of those levels here. And we'll go back to the initiation of translation when the AUG codon that is starting translation, when that's recognized by the 40S subunit in combination with that initiator tRNA MET. Remember, the initiation tRNA is special. It has a MET stuck onto it, and it's brought there as part of what's called a ternary complex. Why ternary? 3 parts: tRNA, EIF2, GTP. 3 different molecules, ternary complex. They come in when the AUG is found, GTP is hydrolyzed, and it's released along with EIF2. And that enters in a cycle to make more ternary complex, and that's done by a protein called EIF2B. It grabs EIF2 and GDP and exchanges the GDP for GTP, and now this can go and join another tRNA and make a new ternary complex. So you can see you have a cycle. You start translation, you release GDP, EIF2, it's recycled, and you start over again. This is a very important part of translation regulation. The amount of EIF2B is highly regulated, and that can regulate translation. A number of conditions in the cell, including ER stress, amino acid deprivation, or the presence of double-stranded RNA, which would be a sign of virus infection. These activate what are called EIF2 alpha kinases. So this blue protein is EIF2. It's a multi-subunit protein. And its alpha subunit can be phosphorylated by one of these kinases. So this phosphorylation, so what is ER stress? Well, you're making a lot of proteins in the ER and they're misfolding for whatever reason. The ER can sense that. It has a protein in the lumen that senses misfolded proteins, and it goes, we're going to shut down translation because we have to figure out what's going on here. Until we can fix it, there's no point in making more proteins. And so in the case of ER stress, there's a kinase called PERK, phosphorylates EIF2. So here is the phosphorylation of EIF2. You take ATP, you put a phosphate on EIF2. Now you have GDP EIF2P. When this goes to be recycled for GTP, it binds EIF2B, but it can't be released. It can't be exchanged because the phosphate makes this complex bind EIF2B too tightly, and it cannot be recycled for GTP. And so what this effectively does is reduces the amount of free EIF2B, so all recycling stops of EIF2 and translation is inhibited. So ER stress, amino acid deprivation. If there are not enough amino acids around, you don't want to start making proteins because you'll miss amino acids and they'll be bad proteins. So again, that sensor GCN2 phosphorylates EIF2 alpha, shuts down protein synthesis at this level. Again, because you can't make new GTP EIF2. Now, double-stranded RNA is made in virus-infected cells, and it's sensed by a protein called PKR, which phosphorylates EIF2 alpha and does the same thing. What's the reason for that? Well, the cell says, I'm infected, I'm going to die to save the rest of the organism. Now, that's the way we look at it, right? So that one cell shuts down translation. It probably will limit virus spread. And so as a consequence, every virus on the planet encodes at least one antagonist of PKR in this whole phosphorylation of EIF2 alpha system. So here's how this works. As PKR is made, it's not active as a kinase. It needs to be activated by double-stranded RNA. And there's a molecule of double-stranded RNA. It's being bound by PKR. And the part of PKR that binds it is this double-stranded RNA binding motif. And then there's a catalytic domain or a kinase domain. And when 2 PKRs bind the same RNA, they phosphorylate each other and they activate each other. So the key here is that you need 2 PKR molecules bound to a single double-stranded RNA. They will activate each other, and then the active PKR will go on to phosphorylate EIF2 alpha and shut down translation. So a virus infects a cell, makes double-stranded RNA, through replication or transcription of both strands of a DNA genome. Double-stranded RNA is sensed by PKR. PKR is phosphorylated, it's activated, it phosphorylates eIF2 alpha. Translation is shut down as a means of stopping virus infection. Now, shutting down translation is a pretty extreme move, and it probably will kill the cell, but as I said, that could probably limit virus spread. So PKR is part of the cellular antiviral response, which we'll come back and talk about later. But basically, viruses infect cells, they induce the synthesis of PKR. It's activated by double-stranded RNA. It inhibits host translation, and the cell will probably die eventually by apoptosis. As I said, there are many ways for viruses to interfere with this pathway, because if they don't, they're not going to make it. This is so powerful, this PKR antiviral pathway, that if viruses don't encode antagonists, they're finished. So here's one example. Adenovirus encodes a small RNA called VARNA1 that prevents the activation of PKR. Here's VARNA1 on the left. That's its structure. So normally, adenovirus infects the cell. It makes double-stranded RNA. PKR binds it, phosphorylated. You shut down translation. But when VARNA is made, VARNA binds one molecule of PKR and one only. So PKR cannot be activated. There's no phosphorylation. Protein synthesis continues. So adenovirus without VARNA1 cannot replicate efficiently because it binds one molecule of PKR, not two, as double-stranded RNA does. Only one. And that way, PKR cannot be activated, and it will not phosphorylate eIF2 alpha. As I said, every virus has to encode some antagonist of this PKR eIF2 alpha system, and here they are. That's PERK, by the way, that transmembrane protein. They're inhibitors of PERK. That's the one that senses misfolding of proteins in the endoplasmic reticulum. So that's antagonized by herpes virus proteins. There are viral proteins that act as pseudo-substrates. What does that mean? So the vaccinia virus K3L, it looks just like eIF2. And so PKR goes to phosphorylate it, and the virus makes so much of K3L that there's not enough PKR to phosphorylate eIF2 alpha. So translation occurs. Then there are antagonists of PKR directly. There are antagonists of RNA like VARNA. We just talked about that as well. There are antagonists of the protein itself. And this is just a partial list of all the viral antagonists of different members of this pathway. This tells you how important it is because every virus needs to have a way to get around it. Our next question. PKR is an interferon-induced enzyme that is activated by what, leading to phosphorylation of what and inhibition of translation. So fill in the blanks. GDP, eIF2 alpha, double-stranded RNA, eIF2 alpha, double-stranded RNA, eIF2B, single-stranded RNA, eIF2 alpha, or maybe none. B. So PKR is activated by double-stranded RNA, and it phosphorylates eIF2 alpha, and that inhibits translation. It doesn't phosphorylate eIF2B. eIF2B binds the phosphorylated eIF2 alpha irreversibly. Another response to virus infection, so one you saw is PKR, phosphorylation of eIF2 alpha, inhibition of translation. Another response is the formation of stress granules. So under conditions of stress, ER stress, amino acid deprivation, virus infection, cells take the actively translating mRNAs, which are shown at the top left here, and they put them into these structures in the cells called stress granules. You can see them as little punctate assemblies in the cytoplasm, and there they are not translated. So the idea is let's put all the RNAs in these granules, shut them down until maybe things get better. So this is, of course, not good for viral RNAs. So viruses, as you might guess, have evolved ways out of this. And they're all shown here. For example, these are some of the components of stress granules, G3BP, TIA1, TIAR, and of course, poliovirus cleaves one of these. There's a protease of poliovirus cleaves G3BP, which prevents the formation of stress granules. The little red circle slash, that means the activity prevents the formation of these granules. Many viruses, as we will see, have to build replication complexes out of various lipids, and hepatitis C, its replication complex includes G3BP, which is part of the stress granule, so it takes it away from the stress granule and prevents its formation. And other viruses as well incorporate components of stress granules, G3BP, TIAR, and others, into their replication complexes as a way of antagonizing. So, in other words, sort of like PKR activation, many viruses antagonize the formation of stress granules, which would otherwise shut down protein translation. All right, enough of translation. Let's talk about metabolism. You make a lot of proteins, a lot of virus particles. You need energy, you need precursors. You need nucleotides, amino acids, fatty acids, energy. You need 4 ATP to make a single peptide bond in a protein. You need tons of ATP. So viruses have to crank up the cell metabolism. And we're going to talk about that a little bit. And here's the first example that this happens. This is an experiment showing increased glycolysis, the utilization of glucose in virus-infected cells. So on the left, we have some plates of cells, either mock-infected, infected with a wild-type adenovirus, or a mutant of adenovirus that doesn't replicate well. And we have time points here. We have 24, 36, 48, and 72 hours after infection. And you can see the plates in the middle are getting yellow. So these plates, the medium, have phenol red in it, which is a pH indicator. So as this medium is getting yellow, it means the pH is dropping. And the reason it is because the cells are producing lactic acid, because the virus has cranked up the metabolic pathways, the glycolytic pathways, of course, which lead to the production of lactic acid. And you can also measure glucose consumption in these cells, and that's the experiment on the right. And you can see in red, the wild-type virus, the glucose consumption goes up, whereas the mutant, no. And that's why the mutant is not acidifying the medium. Many viruses do that, including rhinoviruses. So this is glucose metabolism. Glucose, of course, is the major breakdown of the carbohydrate that you eat. There it is at the top there. There are specific transporters that pull glucose into the cell. They can't just float across the plasma membrane. GLUT13 is a transporter of glucose. It's then pulled into the cell and is converted in the glycolytic pathway eventually to pyruvate and lactic acid. And that's what's happening in the previous experiment. The cells are getting yellow because they're making lactic acid out of glucose. And along this pathway from glucose to pyruvate, you can get more energy made. So this is an energy generating system. And along the way, there are some intermediates which you can shunt off and get more energy as well. And furthermore, you can convert pyruvate to acetyl-CoA, and that can be used to make fatty acids for membranes. And so this is perturbed in virus-infected cells, this glycolytic pathway. So for example, in cells infected by human cytomegalovirus, that's a herpes virus, the infection stimulates the uptake of glucose. It upregulates the glucose transporters and makes a lot of acetyl-CoA. Interestingly, in cells infected with herpes simplex virus, the glucose import is inhibited. And what that does, it inhibits the production of pyruvate, and you get a shunting of existing glucose 6-phosphate in the cell to this ribose 5-phosphate pathway. That is a shunt that is used to synthesize nucleotides, the precursors for nucleic acids. In the overall scheme of glucose metabolism, you can see the shunt there from glucose 6-phosphate to ribose, et cetera, and it goes off and you make nucleotides from it. So herpes wants that to happen. Why? Well, we think the reproduction cycle of herpes is short, so it needs a lot of nucleotide precursors to make its DNA, whereas cytomegalovirus, much slower, but it needs lots of lipid to make membranes, and hence going to acetyl-CoA. So that's an example of how viruses can manipulate these metabolic pathways depending on what they need. Fatty acid synthesis is another point of manipulation. Remember, from the glycolytic pathway, the end result is pyruvate. That's imported into the mitochondrion. It's converted to acetyl-CoA in the mitochondrion. Acetyl-CoA cannot get out of the mitochondrion on its own, so it has to undergo the shunt where it's converted to citrate. Citrate is exported by a TCA transport protein. It's then converted back to acetyl-CoA, and it can either be re-imported into the mitochondria to make more energy, or it could be shunted up to make fatty acids. So acetyl-CoA can go through millennyl-CoA and make fatty acids via fatty acid synthase. In cells infected with human cytomegalovirus, most of the citrate leaves the mitochondria and is shunted up to be put into the fatty acid pathway. And we know that if you inhibit this enzyme, millennyl-CoA, or fatty acid synthase, that reduces the yield of virus because it can't make enough envelopes to make the particles. And other viruses show similar properties as well. They require this shunt to make fatty acids. Viruses also have roles in disease depending on where they replicate. Two hepatotropic viruses, hepatitis B virus on the left, hepatitis C virus on the right. They both replicate in the liver, which of course is the site of glycogen synthesis and de novo synthesis of glucose, gluconeogenesis. And these two viruses interfere with those processes leading to type 2 diabetes. Hepatitis B stimulates the levels of the enzymes involved in glucose synthesis. It needs that for its own metabolism, but the consequence is that the patient has too much glucose, it's insulin resistant, type 2 diabetes. Same thing with Hep C, which causes reduced uptake of glucose. That causes the liver to start making more glucose. The virus uses that, but the consequence is type 2 diabetes. So these interfering with metabolic pathways to benefit virus replication have other consequences on the host as well. You may remember the citric acid or tricarboxylic acid cycle from high school even. That's where I learned about it. And this is where the pyruvate from the glycolytic pathway goes to acetyl-CoA, which then enters this TCA cycle. And it's progressively oxidized to eventually oxaloacetate, and along the way you make energy. You can also make byproducts that can go off to, say, fatty acids and pyrimidine and purine. So viruses definitely impact on this in a variety of ways. So for example, human cytomegalovirus pulls citrate out of the cycle so that it can make fatty acids for its membranes. And if you do that, you're going to stop the whole cycle, take energy production away. So what HCMV does is takes glutamine and pulls it into this cycle at alpha-ketoglutarate, and that can resume the cycle. So it pulls out at the level of citrate to make fatty acids, pulls in glutamine, and you can complete the cycle and make some energy as well. And herpes simplex virus wants to make lots of pyrimidines and purines to make nucleic acids, right? And so it pulls oxaloacetate into that side channel as well. So again, this central hub is manipulated in many ways by viruses. Lipid metabolism is another key place of intervention. This is a triacylglycerol at the top left. 3 fatty acids, which are linked to glycerol. And this is the way we store them as triacylglycerols. And these are our primary energy sources. They can be oxidized to yield energy. And of course, you can store the lipids in this as well, and you can use it to build membranes. And viruses, of course, modulate this not only to have membranes for virus particles or for replicating genomes, but they will oxidize fatty acids to get energy. And the way these are carried around in the body is as triacylglycerols on lipoproteins in your bloodstream. When those are needed to be broken down, there are plasma membrane-bound lipoprotein lipases that break up the triacylglycerols into free fatty acids. They're imported into the cell by specific transporters and converted to acyl-CoA. And they can also be stored in a process that happens in the endoplasmic reticulum on lipid droplets inside of the cell. Now, viruses, of course, manipulate these pathways. Here's an example of human cytomegalovirus. It wants very long fatty chain fatty acids for its assembly. I showed you before that infection increases carbon flux from glucose to acetyl-CoA, and it does so by interfering with the normal pathway for fatty acid synthesis. So the genes for fatty acid synthesis in the cell nucleus are encoded in the DNA, of course. They include all the enzymes you need for that. And the promoter is activated by this transcription factor called SREBP1M. That protein is normally sequestered in the endoplasmic reticulum, all the way at the top there. You see SREBP1P is held in the ER by interaction with two other proteins. When fatty acid levels are low in the cell, that causes PERK to dimerize. That causes a downregulation of this regulatory protein INSIG1. It releases SREB1P, which moves through the Golgi and into the nucleus and activates the genes needed for fatty acid and sterol synthesis. So, for example, if cholesterol is low, PERK will sense that, dimerize, and SREBP will move out and into the nucleus. And of course, viruses can interfere with this to get what they need. And here's an example of that in cells infected with hepatitis C virus. A transcription factor for the synthesis of SREBP is FOXO1. Normally, FOXO1 leaves the nucleus and very little SREBP is made. Hepatitis C virus infection blocks its exit from the nucleus, so it remains in the nucleus where it stimulates the production of SREBP. And as we saw in the previous slide, that stimulates lipogenesis. Now, the other side of the lipid story is oxidation to make energy. And so the cell wants to have a balance of the two, of course. The genes that are needed for fatty acid oxidation are shown down here on the right. They're controlled at their promoter by a transcription factor called FOXA2. And hepatitis C virus decreases the amount of FOXA2 in cells to limit the use of fatty acids for oxidation. It wants them, the lipids, to be made and accumulate for its production. And we know this because if you overproduce FOXA2, you inhibit virus replication because it pushes the balance towards oxidation. And if you knock down FOXO1, you also inhibit replication because you cannot make the lipids that are needed. And at the top right is a staining of cells with a dye that shows the lipid in green. And so these are human hepatocytes in culture. These are mock infected. The nucleus is stained with DAPI. It's blue. You can see there's a little bit of cytosolic lipid, but on the right, much more lipid accumulating in the cytosol in HCV infected cells. So again, the virus is pushing the cell to making more lipids, to doing lipogenesis as opposed to oxidizing these fatty acids. How might virus infection lead to increased levels of ATP, stimulation of glucose uptake, increased glycolysis, increased oxidation of fatty acids, increased utilization of glutamine, or all of the above? It's all of the above, which most of you got. Everything, stimulation of glucose, increased glycolysis, increased oxidation for some viruses, increased utilization of glutamine. All right, the last thing I want to talk about is remodeling of cellular organelles. Viruses do this to produce replication sites. And here's an example for poliovirus. On the top is a staining of cells, again with this dye, BOTA-PFA, which stains for fatty acids. Mock-infected cells on the right, you can see little punctate areas of fatty acids. Polio-infected, a huge increase in the amount of fatty acids in the cytosol. And the reason is that the virus is making vesicles where it will replicate its genomes. On the lower left is an electron micrograph of virus-infected cells. And you can see all these double membrane vesicles. The ER is gone, the Golgi is gone. They have all been repurposed to these double membrane vesicles. And on the surface of those vesicles, the virus replicates its genome. And how that happens is shown here on the right. The ER and Golgi are repurposed to form these small vesicles. RNA synthesis occurs on their surface. These eventually become autophagosomes. And they become double membrane by this curvature mechanism. And virus particles that are captured in them by chance can eventually be released at the cell surface in vesicles or without vesicles by an alternate pathway. So autophagy seems to be involved in the generation of these vesicles. It's stimulated as a consequence of virus replication. And as I said, the complete ER and Golgi is repurposed to form these vesicles, thousands and thousands of them, on which the viral genome is replicated. Another example of this is hepatitis C virus, which also repurposes membranes in the cell, but in a different way. And here on the top left, there's an electron micrograph of these vesicles that arise in virus-infected cells. And on the right is a three-dimensional reconstruction of them. They are derived from the ER, as you can see on the lower left here. The ER pinches off to form these structures in which viral genome replication occurs. You can see the viral polymerase is in there, and it's making RNA copies of the genome. And they may also become double membraned as well. And eventually, this is where maturation of virus particles occur. If you look at the right panel, this is a zooming in on one of these vesicles. You can see that a variety of viral proteins are coating a lipid droplet, again, which has been made in response to virus infection. And in this vesicle, RNA synthesis has occurred. And you can see now at the top, a virus particle is beginning to bud into the lumen of one of these vesicles. So that green is a new RNA molecule that has been made. It's associating with capsid proteins and some envelope proteins. And eventually this will bud off into that space and move to the cell surface. So again, this whole network of vesicles transformed the normal structure of the cell, is made for RNA synthesis, and in this case, assembly as well. And the last example I find is a great summary of everything I've told you in a way. This is a plant virus, tomato bushy stunt virus, where it recruits an enzyme into these vesicles to make ATP needed for RNA synthesis. And so this virus in plants induces the synthesis of vesicles, RNA vesicles made of membranes. You can see that in the middle here. And inside of those vesicles, the RNA synthesis occurs. But what is particularly neat about this is that the RNA polymerase, which is this red protein, P92, binds to pyruvate kinase, PK. And that, of course, is an enzyme that is needed to make ATP. The polymerase recruits it into this vesicle. So now it's in the vesicle there. The polymerase is making RNA. And of course, you need ATP to do this. And there's the pyruvate kinase, there, PK, which is making ATP that's needed for RNA synthesis. I think that's remarkable that the polymerase has evolved to pull this enzyme into these isolated vesicles to make the energy that is needed for RNA synthesis. So that's really just an overview of what happens. There's so many different things that happen to transform a cell into an infected cell, but I think you get the overview from that. And that completes our look at a reproductive cycle. Next time after your exam, we're going to start looking at how viruses cause disease, and we'll talk about infection basics.